problem solving and explanation live session on NPTEL Bioinformatics Algorithms and Applications course. So today we'll be seeing about protein folding and how this folding has been quantized. So we try to understand about folding rate. Then we'll move on to the concept of protein interactions. So we'll be discussing about various protein interactions such as protein-protein interactions, protein ligand interactions, protein nucleic acids, and protein lipid interactions. To start with the protein folding problem. So protein folding is a process by which a polypeptide chain folds it to become a biologically active protein in its native 3D structure. So this native 3D structure is crucial for its function. So this folded state is stabilized by various molecular interactions. So during translation, that is conversion of mRNA to a protein, each protein is synthesized as a linear chain of amino acids or a random coil which does not have any 3D structure. So the amino acids in the chain eventually interact with each other to form a well-defined folded protein. So what determines the structure? So the initial amino acid sequence of a protein, that is the primary structure, determines the 3D structure. So this folding process is crucial for their function. If failure to fold properly, they produce as an inactive or a toxic protein that could malfunction and could lead to diseases. So 4 stages of protein folding has been discussed, which gives rise to the various 3D protein shapes. So the structure of a protein is hierarchically arranged from a primary to quaternary structure. So the wide variation of amino acid sequences account for different conformations in protein structure. First is the primary structure, which refers to the linear sequence of amino acids in the polypeptide chain. Second is the secondary structure, which is generated by the formation of hydrogen bonds between the atoms in the polypeptide backbone, so which folds as either alpha lysis or beta sheets. Next, tertiary structure is formed by the folding of secondary structure helices into one another. So this tertiary structure is the geometric shape of the protein, and it usually has a polypeptide chain as a backbone with one or more secondary structures included. So these secondary structures is determined by the interaction and bonding of amino acid side chains in the protein. Finally, we have a quaternary structure or quinary structure results from the folded amino acid chains in tertiary structure interacting further with each other. So these interactions will give rise to a functional protein. So factors affecting the protein folding, like as we know, protein folding is very sensitive process that is influenced by several factors, including temperature, pH, chemicals present around molecular crowding. These factors influence the ability of a protein to fold into their correct functional forms. Extreme temperatures affect the stability of a protein and cause them to unfold or denature. So we have a concept arising from this is the thermal denaturation. So this behavior of protein or unfolding of a protein under the influence of temperature is used to study the folding and unfolding dynamics of the protein. So this is concept called thermal denaturation. Similarly, pH, mechanical forces and chemical denaturation can denature proteins. During denaturation, proteins lose their tertiary and secondary structures and become a random coil. So although denaturation is not reversible, so once the protein is denatured, the chances of it to renature and become its native conformation are very less. But still, some proteins can refold under certain conditions. Further, we have a concept of heat shock proteins and chaperones that protects protein in a cell against denaturation. So these chaperones help proteins to fold and remain folded under the extreme temperatures, like varying ranges of temperatures. And they also assist misfolded proteins in unfolding and refolding correctly. So here I have first question for today, which is what factors can influence the ability of a protein to fold into the correct functional form. So given here, option one is the cofactors and chemicals, so which are comes under the surrounding environment of a protein. So any changes in surrounding environment could uh, lead to the misfolded protein. Then we have temperature and pH, which are very important for the activity of a protein. So indirectly the activity of a protein again depends on its correct folded structure and molecular crowding. The effect of surrounding biomolecules on protein is also crucial for their final state maintaining. So here all of the above influence. So that protein folding can be influenced by various external factors including temperature, pH, chemicals and environmental conditions such as space limitations and molecular crowding. So here is the second one. What happens to proteins during denaturation? So whether they gain their tertiary and secondary structures, or they lose their secondary or tertiary structures, or they remain in their native functional state, or they become more stable. So denaturation could be taken. Denaturation generally studied using thermal and chemical methods. So what happens during the protein? So what happens to the proteins during denaturation? Whether they gain their structures, lose their secondary structures, or remain intact in their 3D structure, or they become more stable. You can answer using the chat box. I will be having a look at the chat box. Okay, thank you for the answer. Yeah. So in the process of denaturation, they lose their tertiary and secondary structures. That is, denaturation disturbs the native 3D structure of proteins. So previously in the introduction we have discussed the misfolding or not proper. So deviating from the proper protein folding could be the devastators causing several diseases. So here I am discussing about mainly some types of diseases. So misfolded proteins denature easily and lose their structure and function. So this incorrect protein folding can lead to many human diseases. So Alzheimer's is an example of a neurodegenerative condition which is caused by protein misfolding. So this disease is characterized by the dense plaques in the brain caused by misfolding of secondary beta sheets in beta amyloid proteins present in the brain matter. Similarly, we have Huntington and Parkinson disease, which are other examples of the diseases that are associated with misfolding. So here in some of these diseases, we have a particular mutation or a particular insertion or deletion that is available, so that their native structure is deviated, and so by their native function is also deviated, causing the disease. Then we have cystic fibrosis, which is caused by CFTR, that is cystic fibrosis transmembrane conductance regulator. So here we can see uh, deletion of a phenylalanine at position 508 in the CFTR protein will cause the misfolding of this protein. Similarly, several allergic reactions have been shown to be caused by incorrect protein folding. So we have experimental studies to understand protein folding rate, but parallelly we also have computational studies for studying the same. So the field of computational studies on protein folding encompasses various methods and approaches aimed at understanding the complex process by which proteins assume their functional three-dimensional structure. One of the fundamental concepts in this field is Lewinthal's paradox, proposed by Leventhal in 1969. So Leventhal observed that number of possible conformations for an unfolded polypeptide chain is large due to the numerous degrees of freedom involved. If a protein were to fold by sequentially sampling all the possible conformations, it would take impractically long time and much longer than what is observed experimentally. So this paradox led to the relation that protein must fold through a series of metastable intermediate states rather than randomly sampling all the conformations. Then we have energy landscape of protein folding, which provides a conceptual framework of understanding how protein navigates their folding pathways. According to this view, proteins fold into their native structures through a funnel-shaped energy landscape, where native state represents the lowest energy state, as shown in the image. So this funnel-shaped landscape allows the proteins to explore various pathways and intermediates while ultimately converging to a native state. Here, the theory of minimal frustration suggests that natural selection has optimized the protein sequences to minimize the frustration in the folding process, ensuring effective folding kinetics and stability. Computational modeling plays a crucial role in studying protein folding. So techniques such as molecular dynamic simulations allows researchers to simulate the folding process and explore dynamics of the process in silico. So these simulations can provide insights into the folding mechanisms, kinetic and structural stability. However, due to computational limitations in simulation techniques, the requirement of computational power, simulations of larger proteins or long-time folding processes still remain challenging. So we have some computational projects such as Rosetta, Foldit, which aim to crowdsource the computing power for protein folding simulations. These projects contribute to our understanding of protein folding and facilitate the development of new computational methods. In recent years, AI methods have been used in protein structure prediction, such as DeepMind's AlphaFold, which is achieved remarkable accuracy in predicting protein structures, and its performance in prediction has been proved to be good in CASP competition. Yeah, I'm still just giving about AI. I'll change the slide. So AlphaFold structure represents a significant advancement in computational biology. Yeah, so here I have some set of questions which are like, what structure of a protein refers to the local folding of polypeptide chain? So we can take one by one, six are given here. So we are taking the first one. So what structure of a protein refers to the local folding of polypeptide chain? Whether it is primary, secondary, tertiary or quaternary. So the local arrangement of amino acids in a polypeptide chain represents what level of structure organization? So thank you for the answer. So the secondary structure of a protein refers to the local folding, and the tertiary structure of a protein refers to the overall 3D arrangement of its secondary structure elements. Then we have quaternary, which is a combination of multiple subunits to form. So hydrogen bonds are important because these hydrogen bonds between amino acid residues are important for formation of secondary structure like alpha helix and beta sheets. Then we have a tertiary structure. So mostly the denaturants affect the tertiary structure of a protein by changing its environmental conditions. And alpha helix and beta sheets are example of secondary structure. Moving forward, I'm talking about protein folding rate. Protein folding rate refers to the speed at which protein molecule adopts a native three-dimensional structure from a random coil or unfolded state. The process of protein folding involves integrate movements and interactions between different parts of the protein molecule, leading to the formation of stable structure. Several factors influence the folding rate of a protein. So, generally, first one is size and complexity. Generally, smaller proteins tend to fold more quickly than larger ones due to the few number of possible conformations they can adopt. However, folding rate also depends on the complexity of protein structure and number of interactions needed for proper folding. Then we have primary structure, which is the sequence of amino acids. So the sequence of amino acids in protein primary structure determines the folding pathway and influences the folding rate. Some amino acid sequences naturally promote rapid folding, while some may lead to misfolding or aggregation, slowing down the process. The surrounding environment, including factors such as temperature, pH, ionic strength, and presence of other molecules, can significantly affect the protein folding rate. And the optimal conditions for protein folding vary depending on specific protein. And we have other support proteins like chaperone proteins, which assist in protein folding by preventing the misfolding and aiding in the correct folding pathway process. So these chaperone proteins can enhance the folding rate by stabilizing the intermittent structures and prevent aggregation. And next we have denaturing chemicals, which can disrupt the protein structure and slow down the process of protein folding. So understanding protein folding rates is crucial for understanding protein structure function relationship as well as applications in drug design and biotechnology. So various experimental and computational methods such as kinetic studies, molecular dynamics simulation, and bioinformatics analysis are employed to investigate and predict protein folding rates under different conditions. So here we have a few questions on what we have discussed. Yeah, so we'll be discussing about chevron plots. So there, how it is calculated and all we can discuss. So according to the energy landscape model, so what represents the lowest energy state for a protein? According to the energy landscape model, so, so this is the energy landscape model. So what does the lowest energy represent? It represents the native state. So in general, native state is considered as the one with the lowest energy. So how do molecular chaperones influence protein folding? So we have studied about molecular chaperones and factors affecting the protein folding. So how do they influence protein folding? Either they denature or they promote protein aggregation or they prevent misfolding and help in correct folding pathway, or they might disrupt. molecular chaperones assist in protein folding by preventing misfolding and guiding the protein along the correct folding pathway. So with the recent advancements in artificial intelligence has shown a remarkable accuracy in predicting. So which among the following contributes to that? So these are the different projects that have been undertaken by DeepMind and Google. So which among the following is an AI-based advancement that helps for the protein structure prediction? So it is AlphaFold. So AlphaFold uh, is the DeepMind's AI-based project. So there are further several versions of AlphaFold. That is AlphaFold 2, which has very good prediction accuracy than AlphaFold. Then we have AlphaFold Multima, which has very good prediction accuracy for predicting heteromeric, homomeric complexes of proteins. According to Levinthal's paradox, why it is impractical for proteins to fold by sequentially sampling all the possible conformations? Why it is impractical to fold for a protein to fold by sequentially? According to Levinthal's paradox, so here we have discussed about Levinthal's paradox. So here Leventhal observed that number of possible conformations per unfolded polypeptide chain is large due to the number of high number of degrees of freedom involved. So if a protein were to fold by sequentially sampling all the possible conformations, it would take an impractically long time, than which is higher. So because the impractical record, it is impractical due to the large number of possible confirmations. So as we have discussed, protein folding is a dynamic process where a polypeptide chain assumes its functional three-dimensional structure. So understanding the kinetics of this protein is crucial for how the proteins achieve their native state and perform their biological functions. Two-state and three-state models are simplified frameworks commonly used to describe protein folding kinetics, and each offer insights into different aspects of protein folding. So in the two-state model, protein fold is assumed to from the occur directly from the unfolded state to the folded state without any intermediates. So here the folding rate constant Kf represents the rate at which protein transitions from unfolded to a folded state. This model suggests that folding proceeds to a single rate limiting step where phase change delta F. So we have there are many experimental ways to measure the delta G values. So this delta G value, which is associated with folding, determines the folding rate constant. So since we are calling it a rate constant, the units will be per second or per time units. So the value of delta G can be experimentally determined using methods such as equilibrium unfolding experiments. Then we have a three-state model. So this model acknowledges the possibility of intermediate states during protein folding and adding complexity compared to the two-state model. In two-state model, it is like protein folding is assumed to directly occur from folded to unfolded state without any intermediate. So here one intermediate state. So adding this intermediate state increases the complexity. Here in three-state model, proteins are assumed to fold through one or more intermediate state before reaching the final folded state. The overall folding rate constant is described by equation Kf equals to K initial and K intermediate folded, and K initial and K intermediate folded as a function of these. So this model suggests that protein folding involves multiple rate limiting steps where the formation of intermediate plays a significant role. So experimental techniques such as kinetic measurements, stopped flow spectroscopy, and NMR spectroscopy are used to determine the folding rate constants and understand the folding pathways of proteins. So these models provide a simplified descriptions of protein folding kinetics but may not fully capture the complexity of folding in all the proteins. For more integrated folding scenarios like involving multiple intermediate states or non-equilibrium process, more sophisticated models may be required, and which is under current research concept. So the folding rate of a protein can be influenced by various factors, including the sequence of amino acids that make up the protein. In general, proteins that fold quickly tend to have higher proportion of small and hydrophobic amino acids, while the proteins that fold slow tend to have higher proportion of large, polar and charged amino acids. Like there can be many examples that we can. One such example could be in case of just give me a second. So one broad example here is the intermittent states are generally defined by molten globule. So we can take immunoglobulins. So all the immunoglobulins have folding intermediates, that is different structures whose energy is little higher than the native state uh, energy, and these intermediates of immunoglobulin folding tend to have key role in defining the protein folding and stability pathways. And there are some proteins whose transition states or intermediate states tend to have binding affinity with certain cofactors or other molecules, which tend or which make them to acquire a final folded state or functional state. So, starting with the fast folding proteins. So, fast folding proteins tend to have compact and stable native state, which means that folding process is relatively fast and efficient. In general, fast folding proteins tend to have high content of uh, hydrophobic amino acids, which tend to cluster together in protein's interior. So, these hydrophobic interactions helps to stabilize the protein native state and also promote rapid folding. So, one example of fast folding protein is a Wilin headpiece subdomain HP35, so which folds into its native state in just few microseconds. So when we compare with other versions of this Wilin headpiece subdomain, this HP35 contains higher proportion of hydrophobic amino acids, which helps to stabilize and also rapidly fold. Then we have slow folding proteins, so tend to have more complex folding landscape, which means that the folding process is slower and more prone to misfolding. Slow folding proteins tend to have higher content of polar and charged amino acids, which can form complex electrostatic interactions that slow down the folding process. Slow folding proteins are also observed to have longer and more flexible regions, which can adopt a variety of conformations, making the folding process more difficult. One example is enzyme adenylate kinase, which takes several seconds to unfold into its native state. So in this kinase enzyme, which contains higher proportion of polar and charged amino acids, and these are expected to form a strong interactions with the water molecules and other polar molecules in the environment. So these additional interactions with the water and other polar molecules can slow down the folding process by creating a barrier to the formation of the protein native state. However, it is important to note that yeah, as of now, I don't have any proper statistics with me, but fold rate is a server from IIT Madras. So for more experimental data, it's a database and prediction server fold rate, FOLD fold rate, that could help you with so these examples of yeah, faster folding, high rate, less time. Since it is inversely proportional to time, so higher value of folding rate gives rise to less value of time in less time. So here one thing that is observed is the preference of amino acids in fast and slow folding proteins is generally related to its size, hydrophobicity and polarity. So other concept that is studied in research is protein folding and folding rates can vary significantly among different protein structural classes. So here I have taken all alpha, beta and alpha and beta. So the first is all alpha proteins, which are characterized by the predominant secondary structure consisting alpha helices. These proteins tend to fold faster compared to the other structural classes. The relative simple folding pathway of all alpha proteins is attributed to the presence of stable alpha helices that can form early folding process. Then we have all beta proteins, which have predominantly more of beta strands. They typically fold more slower than all alpha proteins. Yeah. So here I have a question that tells micro to milliseconds of folding time. So is this having any relation to folding rate? Yeah, since I mentioned the unit of folding rate is per time, like this 1 per time unit. So for example, if we are having a microsecond folding process, so 1 divided by microsecond will give higher folding rate value, 1 microsecond. So if we are having 1 millisecond, so if we have 1 divided by microsecond, the value is 10 to the power of 6. The rate, folding rate is, since it is 1 divided by 10 to the power of minus 6, so which gives 10 to the power of 6. That is higher folding rate. If you take 1 millisecond, a protein is having 1 millisecond as a folding time. That is 1 divided by 10 to the power of minus 3, since it's millisecond. So when we take it to numerator, it is 10 to the power of 3. Yeah, okay. So this is how different. So despite this general trends that is observed in different folding rates of different class of structural class of proteins, there is a significant variability within each structural class. So individual proteins within same class may exhibit unique folding kinetics due to differences in sequence, size and specific structural features. Uh, Further, it is also studied that factors such as presence of disulfide bonds, post-translational modifications and interactions with ligands and other molecules can further influence the folding rates. So here, so which types of amino acids tend to characterize fast folding proteins? So with our understanding that that presence of interactions with other ligands and other molecules can further influence folding rates, we further will be entering into the concept of protein interactions. So which type of amino acids? Yeah. In general, fast folding proteins tend to have high content of hydrophobic amino acids. So what happens if you have a high content of charged or polar residues is that they can form strong interactions with the surrounding water molecules and other polar molecules in the environment. So these interactions can slow down the folding process by creating a barrier to the formation of the protein's native state. So moving on to the next one.
Is it okay to talk?
Yeah, please, please.
Okay. When I am saying you have a protein having alpha, you said alpha structure, it folds faster. When you say beta structure, it folds slower. Is there any structural uh, rationale? See, first primary structure is there. It will form, let's say, alpha helices, and there is a turn or a loop, then another alpha helices, then a turn or a loop, another alpha helix. And when they fold, probably already alpha helices are there, and then they come into a compact shape. That is what is happening. In case of beta structure, you have elongated beta chains probably, not yet formed any kind of hydrogen bonding, and maybe there are residues for turn or turn initiators, and it will start forming either parallel or anti-parallel beta shape, and you say that it is folding slow. So what kind of rationale we can give? Not the statistics of observed thing. What exactly is happening with the structure? As you can see in the alpha helices, it is not going to form any kind of bond breaking from the solvent from the alpha helix to another alpha helix, which will interact directly, not much of solvent interaction. Whereas in the case of beta structures which are extended, there is a lot of solvent in and around the peptide bonds. It has to be replaced, I mean, it has to be displaced by another beta sheet structure to form the hydrogen bond. And these solvents have to form their own uh, hydrogen bond, which requires a little bit of entropic character, which may slow down. So is there any paper in and around this simulating why alpha helices fold faster, why beta structures fold slow? That's what I was just trying to
Yeah.
tell you.
So I hope you have answered, like you have a question and answer itself in your statements. So when we come to the presence of alpha helical content and beta sheets and their effect on folding state, The work on this is started from late 1950s in that. So there was a book proposed by a gentleman about the folding states of globular proteins, that is understanding the molten states in globular proteins, and as well as their comparison with membrane. So they have particularly focused on alpha proteins. By that time, they didn't study anything about beta protein. So their 10 is the exposure of solvent and the presence of residues around the environment around the alpha. So the interresidue interactions in alpha, alpha proteins and beta sheets are the reason for their differences in folding state. So were the parameters of late, I don't remember, around 2008 or 2012, there has been studies showing that how the long range interactions, the short range interactions and the medium range interactions in same different class of proteins, all alpha proteins, all beta proteins and other two classes of proteins affect their folding rate and stability. So it all started based on their analysis of environment. Yeah, so as we discussed, the presence of large and polar residues could hinder in the folding of a protein, so they will have a class of low fold. So, since we have studied the interaction of proteins with other molecules is crucial for its folding, so first I will be trying to define protein-protein interactions, which refers to the physical contact between two or more proteins leading to the formation of complexes or assemblies that perform various cellular functions. So here the top image shows one subunit of protein, that is subunit 1 interacts with the second subunit 2, forming a protein-protein complex. Here I have highlighted the interface, that are regions which are close to its binding component are generally termed to be as interface residues. So there are several types of protein-protein interaction, that is stable interactions and transient interactions. So when we come to stable interactions, so these interactions are long lasting and essential for the structural integrity and function. So these stable interactions are often observed in structural scaffolding, cellular organization and maintaining the architecture of macromolecular complexes. Examples include uh, examples include within the ribosome, DNA polymerase or proteasome. Then we have transient interactions, so which are relatively short-lived and dynamic, and they occur only to regulate a specific cellular processes. So these protein-protein interactions are often highly specific with proteins for selectively binding, they're like highly specific with proteins selectively binding to their interacting partners. And these protein-protein interactions can exhibit varying affinities ranging from weak to strong interactions depending on the strength of binding forces involved. So methods for studying protein-protein interactions experimentally, we have yeast 2 hybrid assay, co-immunoprecipitation, or other methods. Then we have protein nucleic acids interactions, which are involved in binding of proteins to specific region or sequences within DNA or molecules, DNA or RNA molecules, resulting in the formation of protein DNA or protein RNA complexes that perform diverse biological functions. So here when we observe protein nucleic acids interactions, we have sequence specific interactions and non-specific interactions. So these non-specific interactions occur when proteins bind to nucleic acids in more general or a non-specific manner without any strict sequence specificity. And we have sequence specific interactions where proteins recognize and bind to specific nucleotide sequences within DNA or RNA molecules. Then we have protein lipid interactions. So mostly these are observed in case of membrane proteins or membrane associated proteins. So these are useful for different cellular processes such as membrane trafficking, signaling, and maintaining the membrane structure. So then we have protein small molecule interactions. So this the other name could be protein ligand interactions. So which wherein one try to understand the physical contacts between proteins and small molecules which play roles in regulating protein function, cellular signaling, and other. So So we have known that the complexes exist. The first and foremost thing to understand is how the interaction happens. To understand the interaction thing, first we need to identify the binding sites. Still again, having a complex experimentally derived protein complex structure, it is very easy to identify the binding site. Still, so we have three major approaches for identifying a protein binding site when its complex structure is available, whether it's computationally predicted, like protein-protein complex structures can be predicted using several docking algorithms. So they are distance-based methods, accessible surface area methods, and energy-based methods. So starting with the distance-based methods predict the binding sites based on the spatial proximity of protein residues to the known ligand binding sites or molecules. These methods identify a region on protein surface that are within a certain distance threshold from ligand or a binding partner. So we have algorithms like pocket finder algorithm, CASP, to predict this. So what happens in a distance-based molecule is using the Cartesian coordinates that are given in any PDB file or a structure file, they try to find out the molecules or residues of a protein that are within a required cutoff. So these cutoff values may depend based on different types of proteins, different types of bio interacting partners we consider. So for example, we define a cutoff of 6 angstroms. So the residues that are within 6 angstrom distance of this binding partner are turned to be binding site residues. So this is a distance-based method. So these distance-based methods are relatively fast and computationally efficient and suitable for identifying pockets or cavities on protein structures. But these may miss binding sites that are not located within a well-defined pockets or cavities and limited ability to predict the binding sites in a flexible regions of proteins. Then we have accessible surface area based methods. So accessible surface area based methods predict the binding sites based on the solvent accessible surface area of protein residues. So here uh, we have a get area method or DSSP base method. So given a complex molecule, first calculate the surface area accessibility to a complex. So residue 1 has X surface area, X1 surface area, surface accessible, solvent accessible surface area. Residue 2 has X2. Similarly, take the individual molecules and calculate their solvent accessible surface area. So in the two cases, in their unbound state, in the bound state, if two residues is having same solvent accessibility, then the chances of it to be in the binding site is less. So what happens is, when we see the differences in their solvent accessible surface area in the bound state and unbound state, if there's a difference, then the chances that these residues are in binding site is high. So this is how the accessible surface area method works. Then we have energy based method. So they typically involve computational docking or other methods to calculate the binding energy and identify the favorable binding sites. So we have examples of autodock, gold, other things. Considered more accurate. So energy-based methods are considered more accurate in predicting the binding sites and binding affinities, and can capture detailed interactions between proteins and other its binding partners. But it has its own limitations, that is computationally demanding and time consuming, and requires high quality protein structures and may be sensitive to structural inaccuracies. So in summary, each method for predicting the protein binding site has its own strength and limitations, and often choose a method or combination of these methods based on the specific requirement and constraints. So amino acids exhibit different binding propensities depending on type of biomolecular interactions, such as protein-protein interactions, protein DNA, like protein nucleic acids, or protein ligand interactions. These propensities are influenced by various factors, including the chemical properties of the amino acids. Understanding the binding preferences is crucial for designing effective drugs and therapies targeting the specific interactions. So in protein-protein interactions, hydrophobic amino acid residues are commonly found in protein-protein interfaces due to their ability to form hydrophobic interactions with other hydrophobic residues. And we also see hydrophobic amino acids present in the binding interface contribute to pi-pi stacking and other and hydrogen bonds. We also have charged amino acids that can form salt bridges and electrostatic interactions. So in case of protein DNA interactions, basic amino acids such as lysine and arginine are commonly found in DNA binding proteins due to their ability to form electrostatic interactions with the negatively charged phosphate backbone of DNA. Then we have protein ligand interactions. So protein ligand interactions are predominantly have different types of interactions based on the nature of the ligand that we use. So I have few questions for today. So protein folding rates provide information on. So protein folding rates provides information on how fast it assumes its native structure. So how fast or slow a protein can unfold is depends on its stability value. How fast it assumes its native structure is a protein folding rate. So Here we'll come to introduction of a concept called chevron plots, so which are graphical representations used in protein folding kinetic studies. So they are named after the characteristic V-shape pattern observed when plotting a rate of reaction against concentration of denaturant. So here denaturant doesn't mean a chemical. Temperature is also a denaturation factor that is used. So these plots are particularly useful in illustrating the mechanism and energetics of protein folding and transition. So in the experimental studies, the protein of interest is subjected to increasing concentration of denaturant, and the rate of folding or unfolding reaction is measured at each condition. So then on the x-axis of a plot, the concentration of denaturant or the temperature value is plotted, or on the y-axis, the observed rate constant for folding or unfolding is plotted. So the data points are collected for, yeah, I have a question, just then after solving the question, I'll show you the plot. I have a question on plot, so I'm not seeing the plot, showing the plot. So here is the chevron plot. So kindly answer this question, then I'll move on to explanation of the plot. So for a two-state folding process, this plot typically exhibits a V shape, that is reflecting the transition between folded and unfolded state. So the rising limb of V represents, that is, which shows Kf in H2O, reflects the folding limb, and the falling limb represents the unfolding arm, where increasing. So in the rising limb, that is in the folding arm, we observe increase in denaturant or temperature reduce the rate of folding, where in the falling limb or in the unfolding arm, where increase in denaturant concentrations or temperatures accelerate, that is increase the rate of protein unfolding. So the point where folding and unfolding arm intersect is known as uh, folding midpoint, and that is critical concentration of denaturant that is required, that is denoted by Cm. So the slope of folding arm, that is M, that is slope is denoted by M, Mf for folding, Mu for unfolding. This provides insights into the rate limiting step of a protein. Similarly, the slope of unfolding arm indicates the rate limiting step of protein unfolding. So deviations from ideal chevron shape may suggest that presence of intermediate states or a behavior that is different from a two-state folding behavior. So these are tools used for investigating the folding kinetics and energetics of protein. So particularly those undergo two-state folding transitions. Now, why two-state transitions are stressed? Because till date, for many cases of proteins, the multi-state transitions has been not defined clearly. Yeah, in my knowledge, The three-state or four-state folding of membrane proteins has been recently proposed in 2023. Yeah, before that, still several doubts and question marks were existing. So here, the chevron plots provide a visual representation on how folding and unfolding rates change with varying experimental conditions and offer insights into a underlying mechanism of protein folding. Yeah, so as I mentioned, so they helps in understanding protein folding kinetics. So that's all I have planned for today. Sorry, one more thing I forgot to mention is why a denaturing concentration and the H2O. Why one has to measure, yeah, I'll do it. Why one has to measure any value like uh, rate of folding or rate of unfolding with respect to zero denaturing concentration, because so this zero denaturing concentration they refer to a native state. So none of these values of zero concentration, so where you see these lines meeting the x-axis, y-axis that is 0, we cannot determine these values experimentally. So we use a linear extrapolation method. See, these are the points. Blue color dots are the points that are experimentally determined, and these points are extrapolated such that they meet y-axis, and these values are considered as native state values. So this is the linear extrapolation method. So in this plot, different denaturing concentrations and the observed rate of folding or unfolding is plotted. So with these plots, we backtrace to value which corresponds to the zero denaturing concentration. So these values are termed as a rate of folding or rate of unfolding. So the rate of folding and rate of unfolding, these two curves will depend based on the starting state of the experiment. So the point of intersection of these two points is termed as CM, that is critical concentration of the protein, and the corresponding temperature value we observe is also called as a transition temperature or melting temperature of that protein. So these values have been experimentally determined at different denaturing concentration to observe rate constant at zero denaturing. So this is the plot. So you experimentally observe folding rates at different concentrations, backtrace them, then get the value at zero denaturing concentration and term it as a native state value. This is how the values are calculated. That is the purpose of this plot. So that's all I've planned for today. Any questions I can take? Yes, so it is interesting really. The folding and unfolding rates will be different because the path it takes, like folding to unfolding path and unfolded state to folded state path is not same. So for example, we take a, yeah, so don't go with the orders of magnitude in this plot. This plot has been made to incorporate two. So the orders of magnitude may depend on size and complexity of the protein as well as uh, other other environmental parameters. And sometimes the chaperonin molecules helps to cross the energy barrier. So while unfolding, we may not observe something helping it to regain that energy barrier again. So there are several additional support molecules that helps in protein folding. And while unfolding, one may see many intermittent stable states. Like from global minima, we observe many local minimas that are possible, local minimum energy states that are possible while unfolding.
Sir, one small question. Maybe I forgot to ask. When we say this folded and unfolded, is the secondary structure completely destroyed or it is still there? Some portion of the secondary structure, like alpha LH, it has to be completely destroyed for the unfolded state, or it can be there.
So in denaturation experiments, I'm not sure about globular proteins, but it tells that around 50% of the secondary structure states still be intact in transmembrane proteins. So unfolded states corresponds to not a uh, linear chain, but a protein structure that has some localized arrangements of like alpha helices or beta sheets intact. I'm not sure about values of globular proteins, but in transmembrane proteins, it is observed that denaturation experiments still have nearly up to 50% of their secondary structures intact.
Sir, one more point. When I said the order, the Ku, that is the unfolded constant, is 10 power minus 5, and Kf, the folded state, is around 10.3 or so. So around 10 to the power of 8 is the order for these constant. That's what little bit surprised me. That's all.
Yeah, so observe these are observed rate. So as we know, the proteins tend to uh, fold faster, and for unfolding, they need extra additional sources and effect of those sources in experimentally determining these values could vary. If no other questions, everyone are free to leave.